professor with the Department of Crop and Soil Environmental Sciences at Virginia Tech. And we're going to kind of change gears this afternoon and move away from the methodology and into some case studies and spend the rest of the day on that and begin the day tomorrow with that. So without further ado.
Okay. All right, thank you, Kevin. Let's get everybody in from out where the goodies are. See, it's late afternoon. You've had a lot thrown at you. Learned anything today?
Yes.
Well, certainly, in my 4 colleagues today, Jody, Orrin, Don, and Mike, you have heard the very latest in the current status of microbial source tracking, the methodology, and all of the many decisions that you have to take into consideration when you're deciding to use source tracking. So, what I'd like to do in this presentation is, first, we're going to largely now move away from the methodology. How you're going to use source tracking. You take it out into the field. What I'd like to go over is then what happens. Many of you here are involved in case studies, and as I go through some examples, if you have something to add, some experience that you've had that is germane to this topic, then by all means, please raise your hand and chime in. That'd be most welcome. When I was invited to come down and participate in this conference last fall, my first thought was, well, how hard is this going to be? You can't do source tracking if there's no water. But so if there's no runoff, then what's the big deal down here? So actually, that's an oversimplification, and I wish all case studies were that easy. So in trying to avoid preaching to the choir, my thought was, what can I do to summarize kind of the current status of taking source tracking to the field in case studies, and what are some of the lessons that we've learned that might be of interest to you as an audience, okay? So I thought I would start with a plug on our book, since that's a shameless plug, but Mike did that earlier. There's only 2 books out. Oh, just came in. This came out last summer, and of the 26 chapters, 8 of them deal with case studies. And I've selected 3 of those to use as examples. And the reason that I'm doing this is because most of these case studies that were in the book, this was the first time they've ever been published. It is difficult, and it's been my own experience as well, to get case studies in the peer-reviewed journals for a variety of reasons. Often, you don't have control over the sampling regime, or when and where and how samples are taken. There are a lot of factors that make it difficult to get case studies published in the peer-reviewed journals. And so these studies that I'm going to go over in these chapters are work that hasn't been published before, and I think there are some lessons in it for all of us. Plus, I wanted to use these chapters because the very high-quality scientists who were the authors. This book has 26 chapters, 71 authors and co-authors. and as lead editor on this book, trying to ride herd on 71 independently minded scientists that are just as independently minded as my 4 colleagues here, talk about herding cats, or trying to herd cats. This kind of thing you do once in a career, and I've done mine. But regardless, it was a labor of love, and actually, Jody, those monthly royalty checks are pretty sweet, aren't they? Yeah. You're not getting yours? Uh-oh. Okay, move along. Let's start with chapter 20 on beaches and coastal environments. This particular chapter described 2 case studies, both at marine beaches, one Huntington Beach in Southern California, and it was largely Allie at Stanford and Chris at NOAA who were responsible for that, and a smaller beach on the Atlantic side of Florida, not far from Miami, and Helena and Troy were largely responsible for that. Both of these beaches were mainly impacted by non-point sources, and over time a variety of biological, chemical, and physical methods have been done to try and determine the sources. Let's look at Huntington Beach first. Now, this is southern Orange County, some of the most highly priced real estate in the United States. Long beach season, heavily used. The man hours, or I should say person hours, resources, Squirp has been heavily involved in this beach, and the amount of work and money involved in this beach has been prodigious. It's a very big number. Used a variety of different methods, some of which you've heard about today, most of which went through the process that Don described of listing priorities and then trying to eliminate those one by one, and in this particular case went through that same type of scenario, eliminating Calvert Marsh, the Santa Ana River, groundwater discharge, wastewater outfalls, animal sources, sediments, and sands. So by process of elimination, they eliminated everything. And by eliminating everything, the answer is still the main source of bacterial contamination remains unknown. And it has not been from lack of effort. I don't know of another case where a single beach has received as much effort as this particular beach. Incredible. And the answer is, don't know where it's coming from. Some human is present. Well, duh. Some dogs. Migratory and shorebirds are seasonal, but there's no smoking gun. So this is a quote at the end of the chapter. The findings have taught stakeholders and researchers that contamination of urban coastal waters is a complex and difficult problem to solve. Took a lot of money to come to that conclusion. All right, let's move to a smaller, easier, simpler beach. This is Hobbycat Beach in Florida that Helena and Troy worked on. Less complicated. It's on a small island, Virginia Key, variety of human, animal, and gull markers were tested, Enterococcus species distribution, also tried measurement of bacterial and mainly bacterial pathogens. Went through the same type of process of elimination, eliminating shoreline sand. They noted that some sources were added to shoreline sands. Residual times could be substantial. Direct and indirect contaminant contributions from gulls, dogs, and humans. Wastewater outfall was explored. Didn't receive nearly the amount of resources that Huntington Beach did. But still, this was no small effort. The answer is, main source of bacterial contamination remains unknown. Dogs are an issue, but cannot explain the consistently elevated counts. Migratory shorebirds are seasonal, no smoking gun. Prolonged persistence due to regrowth in the sands. Well, of course, since these are marine systems, the indicator's Enterococcus, not E. coli. But this is something that nobody wants to hear, and Mike presented that this morning or this afternoon, presenting that information on naturalized E. coli that are simply there, living in the environment. Well, maybe that's the same situation on these two beaches. Are there naturalized enterococci that are simply residual in that environment? And no amount of effort is going to key them out to a source where you can find that source and correct it. I don't know the answer to that, but that's certainly now, when we started doing these types of studies, that was way out in left field. And as other options have been eliminated, that is coming closer and closer and closer and closer by process of elimination. So in this particular chapter, these two beaches didn't have a good conclusion on both cases. After tremendous effort, the sources of the bacteria resulting in those persistent closures remains to this day unknown. The next chapter, a little closer, case studies of urban and suburban watersheds. And my colleague Jody is one of the authors on this chapter. And so if you want to update anything or chime in here, Jody, please do so. This chapter described something many of you are familiar with, a weight of evidence approach that allows source tracking methods to be highly focused and used only on an as-needed basis. Involves categorization of sites by microbial water quality assessment and uses a weight of evidence approach along with microbial water quality assessment to determine hot spots and where and when is source tracking actually needed. As Mike mentioned, a lot of times this is the very best source tracking tool you have, but that means you have to be out there to use it. In this particular chapter, a priority ranking of most probable source categories and recommended management options were developed for each sampling site. There were 6 subbasins in the Hillsborough River watershed that were used as examples for the weight of evidence, that should be a WOE, a weight of evidence approach. And the 10 watersheds in Florida were used as case studies and one near Tampa Beach as an example. In this particular chapter, there are a couple of things that I chose that I thought would be pertinent for our discussion here. This chapter emphasized how important local knowledge and agency buy-in are essential for project success. Most of us, my colleagues and I, working in academic institutions, you can't expect us to be that heavily involved in these field studies where we're going to be able to give it the amount of time and effort that it deserves. Local knowledge and agency buy-in is absolutely critical. Some sources are obvious, many are not, and it can take a lot of time and sampling to trace sources to specific points of origin. Another lesson to be learned from this particular chapter is that one small cross-connection, faulty lift station, these were some of the particular sources of pollution that Jody found over some years working on the Hillsborough River, or chronic storm sewer overflows. Even though these are small, if they're persistent, they can impact a large area. And in this particular watershed, there's been a high success rate in finding those sources. This chapter also pointed out something that I've already mentioned. There are not many situations where a source was found, some change was made to address that source, and then subsequent monitoring was done to determine did attempting to fix that source actually fix it. Because that's the bottom line for did source tracking work. Did it lead you to a source? You were then able to address that source, and then subsequent monitoring after that remediation, whatever that was, demonstrated that you had remediated the right source and those indicator bacteria from that source were no longer present. Well, there isn't a good record in the literature of those types of examples, at least not so far. Then the third chapter, agricultural and rural watersheds. This involved two case studies. The first one was an alpine karst groundwater spring system in the Austrian Alps, and the second, a surface water system in Texas, Lake Granbury and Buck Creek. I think we can skip the karstic spring study because it doesn't have a whole lot of relevance to Texas in our conversation here. Although I will admit that I did take a trip and go over and visit Andreas just to make sure he was doing this right. So, Lake Granbury, Texas. Highly developed landscape, sanitary sewers indicated human sources would be a major component. The sanitary survey noted older housing developments and man-made coves that were put in prior to current septic regulations as potential problems. A variety of different methods were used. Methanobrevibacter, human polyomus, Bacteroides, Genbac, a couple of library methods. Keep this thought in mind. Sanitary survey indicated human sources would be a major component. The results. Data indicated that Lake Granbury was impacted primarily by wildlife and livestock to a lesser degree, with only a minor human allocation. Okay. Sometimes source tracking results don't give you what you expect. So in this particular case, why not? Well, here are some possibilities. Were the source tracking results wrong? In other words, did George screw up?
Joy and Elizabeth.
So if we rule that off the table, then if you assume that the results were wrong as being unlikely, remember, weight of evidence, a variety of different methodologies, agreement among methods. When you get agreements from multiple lines of evidence, it dramatically increases your confidence in the results. That's one reason we're still using a toolbox approach. You've heard before, my experience has been the same. There is no one good method. And deciding which ones to use takes a lot of experience and guidance, and it's quite a decision process. So if we rule that out, okay, insufficient sampling over time for a large watershed. Is that a possibility? Is the human pollution staying in the man-made coves? Is there little mixing? Was the sampling regime not sufficient to account for a large percentage of the pollution being human from these man-made coves? Were the wildlife bird populations larger than expected? Was subsurface flow from wildlife or other sources impacting the lake? Subsurface flow from groundwater? Are fecal bacteria carried by fish in the lake? Other possibilities that may impact the results. Suggestion, maybe use intensive targeted sampling to identify hot spots before using source tracking. But to do that right, over seasons, over different impacts on the watershed, are those homes all occupied year-round? Are they seasonal? Are people there at some times and not others? Is it highly influenced by precipitation? Well, to get to that point, it could literally mean hundreds of samples, and hundreds, and hundreds, and hundreds, and hundreds, and of course, a tremendous budget in order to tackle a lake and a watershed of that scale and try and determine where the hot spots are on a body of water that big. Let's move to a simpler one. Buck Creek, the Red River Basin, primarily a rural and agricultural landscape. Row crops, grasslands, relatively undeveloped, ranches and livestock, sanitary survey indicated primarily wildlife and livestock. Many of the same methods, a combination of library independent and library based. Results indicated Buck Lake was impacted primarily by wildlife and livestock as expected, further giving you confidence in the source tracking results. But also had a human allocation that was unexpected. Where's that coming from? Well, in my experience, the library-based methods tend to overemphasize the human contribution rather than underemphasize it. So we've already ruled out that George didn't mess up, right? So if that's unlikely, insufficient sampling over times and seasons and storms, and as some of our colleagues, especially down in Australia, have found, are there human markers and isolates carried by some wild species that they may not have picked that up from humans? It's just a coincidence that the DNA segments that match those human markers are close enough that you get a match. So, suggestion for here. A watershed with low ambient levels of fecal pollution, abundant wildlife, tough to pin down the human source. It would seem to me one approach here, lots of samples from wildlife that may harbor, if not human isolates, may harbor human markers. A possibility. Okay, George, anything that you'd like to add since the book came out last summer about these two?
And definitely in one particular stretch of the Buck Creek study was basically isolated from humans. It had no houses, nothing around at all, and it was gated, locked. And we did find human Bacteroidetes marker, which was very surprising. Based on analysis of wildlife fecal samples, we did find a few wildlife animals harboring the human Bacteroidetes bacteria, or something that cross-reacts at least with the marker. So that was most likely due to a rare occurrence. In this case, very unusual because we knew there weren't any humans around, and that's why it really caught our eye. But we can occasionally find animals.
Yep, okay. But unexpected results. Yes.
What animals?
We had porcupine and I can't. Do you remember what it was?
Rabbit and white-tailed deer.
And that's in Lake Granbury. We had rabbit and white-tailed deer, which come in close contact with humans in the Lake Granbury area.
Usually by way of pools and areas.
Yeah. But we had other animals, porcupine and
Possum? Did you have any possum?
I don't think it was possum.
George, I believe it was coyote and raccoon.
There, we had that also in Lake Granbury, but I know we have an
Okay.
unusual wildlife population.
Okay. Okay.
Yeah.
All right. Well, we'll leave the book here in just a second. Two more chapters I'd like to draw your attention to. Chapter 16 by Peter Hartel on minimizing source tracking at all costs. Once again, emphasized the importance of local knowledge and using source tracking only when absolutely necessary. And in that particular, with Peter and his team, most cases he felt could be solved without resorting to source tracking, but they were relatively simple, straightforward situations to begin with, okay? The trade-off is that large numbers of samples are needed and it can be very, 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 very labor-intensive. Last one, source tracking in Australia and New Zealand. by 3 of our colleagues down there, 12 case studies, all expertly done. This is a crackerjack group of scientists down there. The methods evolved over time from library-based to library-independent. The Aussies, like our colleagues in Europe, they like and use the fecal stanols and sterols in their ratios, even though they've never really caught on here in the U.S. The lesson here, and it's one that Mike alluded to earlier, they found regional differences in their markers, a warning for the rest of us. And Mike's reported this on his box PCR markers for geese and waterfowl. They work fine for the migratory waterfowl that use the central flyway. But for the migratory waterfowl on the west and east flyways that go up and down the coast, those markers don't work well. And so the lesson from Mike's work was you can use his approach, but you need to find your own markers. Well, is that right, Mike?
Yep.
But you're trying to decide which of these markers to use. That isn't what you want to hear. That in some cases you might have to go looking for and develop your own markers. But regardless, at this point, where we are in the evolution of source tracking, there it is. So I'll close here with just a couple more slides of my own experiences and a couple of examples. This particular study was a small agricultural watershed in Northwest Virginia, and we were involved in that watershed on an 8-year study. And all I want to point out, I've never published this, and yeah, I guess I really should, but at this point in my career, now I don't know if I will or not. What I'd like to point out, and this was done in fecal coliforms. Virginia moved to E. coli in 2004, but we were using fecal coliforms when we started this project in 1997. And between 1997 and 1998 is when our Virginia Department of Conservation and Recreation came in with money, worked with the growers to get the cattle out of the stream, fence, plant all the prerequisite species to make the appropriate distances for riparian buffers. I mean, really did it right. This was a case where we were able to go in, put in pasture waterers, karst topography, so you've always got groundwater and unexpected things going on, but do it right. Get the cattle out of the creek, use the proper setback distances, put in the riparian vegetation, let it grow, and then measure what happened. And in one particular site where it was a large farm here at PB12, a large farm with a very large number of cattle, several hundred cattle, and the small stream flowing right through the pasture. It was their only source of water, and in summer of 1997 was before BMP implementation, and we averaged over some 42,000 fecal coliforms. Then after implementation, it began to come down. In Virginia, our old fecal coliform standard was 400. Which would be right about in here. Well, it took that site to drop down below, after the cattle were kept out of the stream, to drop down below the regulatory standard. It took 7 years. So there was a reservoir there of fecal coliforms, and it took years for that reservoir to eventually dry up, drain out, die off, before that site went down below the regulatory standard. It took 7 years. And some of the other sites are bouncing around that standard. Well, what's happened is over the years that riparian buffer vegetation has grown up. Small trees, brush, vegetation. It's great looking. But in an otherwise highly agricultural area, what's happened on that green belt around that stream? Wildlife's moved in. Lots of birds, deer. The deer are out, run around all over those fields at night. But during the day, they're holed up in that green zone because there's cover and there's water. And they feel secure in there. And as those green zones have attracted wildlife, the counts are starting back up again. Yeah, it's not cattle, but it's above the standard. Hey, Virginia's under consent decree. So, before Mike jumps up and down and says, well, who cares? It's a small stream. Nobody's recreating in it. Nobody's getting sick from it. So what the heck is the risk? Well, he's got a point, but we're one of the states that borders Chesapeake Bay, and a number of environmental groups sued EPA for not enforcing Chesapeake Bay regs. And as a result of that legal action, Virginia and other states that border the bay are under consent decree. So we're held to that regulatory standard. Doesn't mean whether the body of water is suitable for recreational uses or not. We're held to that regulatory standard. End of story. There is one silver lining around that cloud, though. I'll never run out of work. So, because that consent decree means that the state has to spend the resources on it to meet those Chesapeake Bay benchmarks. Doesn't matter what the state's economy is. Doesn't matter if we're in a recession or not. State has to spend that money spend those resources to meet those Chesapeake Bay benchmarks. So, my goal in presenting this particular data was to show that once you have implemented a BMP, you may not see immediate success. How many labs, agencies can afford 7 years of monitoring post-implementation to see what happens? How many have that kind of patience? Okay, next to the last slide, you don't have this one. I put it in after I listened to this morning's presentations because I wanted to make another point. And these photographs are courtesy of another colleague of ours, Dr. Joe Lipo at the University of West Florida. This was a small embayment closed to shellfish harvest. It's tidal. It's about a total body of water about 25 acres. A brackish shellfish bed impairment, completely impaired, 100% closed to harvest. Source tracking told us that there was human involved. We did intensive sampling, used optical brighteners as well as intensive sampling, and it told us that there was a particular stretch of shoreline, but because of tidal fluctuations and currents, we couldn't get any closer than that stretch of shoreline. And you stand there looking at that stretch of shoreline, and you can't see anything that would indicate to you where the heck that human contamination was coming from. So Joe had the bright idea of going out there in the winter, because when you're looking for septage, coming out of a septic tank, it's going to have a higher temperature than ambient. There's anaerobic fermentation in that septic tank, and it's got a pretty warm temperature. So you can look for these heat signatures in the winter. And coming out there with infrared detection, we were able to find right here a little hot spot that was maybe about twice the size of the top of this podium. That was it. That one little spot. And that was coming from a 2-family apartment where the connector between the septic tank and the drain field had come apart. Preceded the modern septic regulations, never should have had a drain field in that sandy soil with a high water table anyway. But it did. So when it was hooked up, it was probably leaking like a sieve anyway. But with that, knowing what to look for, then local officials were able to find that with dye testing and remedy it. This hot spot disappeared, and that embayment now is open to shellfish harvesting again, but it took 3 years for those populations to clear out before local agencies opened it up to harvesting again. So even there, once you cut the source off, it took 3 years for it to clear out. But the main reason I put this slide up is to show again that it can be a needle in a haystack approach. One small persistent site, if undiscovered, can contaminate a large area. Okay, final thoughts. That wasn't a good idea. For somebody as close to retirement as I am, that sounds too terminal. Okay. In conclusion, don't use the word final. By now, we understand how to do sanitary surveys, how to do intensive and weight of evidence sampling, how to account for seasonality events, do watershed characterization, prioritize what potential sources are. I'm reiterating the plan that Don presented earlier. Develop a cost-effective plan. I don't know about that one. Selecting the source tracking tools needed. Well, you heard 4 experts this morning explain how to go about doing that. Although Mike's about ready to throw out the baby with the bathwater, and if the genomics approach works. It's too early to say, but if the genomics approach works, I do admit it would be nice for all of us if we could scrap our PCR equipment. But too early. Okay, so what's needed? Well, technology transfer, that's what we're doing here. We're still developing and testing source tracking tools, and certainly in Oren's description of qPCR, the current squirt method study, when will this end? I have no idea. If we're going to use these markers, we probably need some kind of a microarray approach. If the genomics approach works, another couple of years, we may be moving away from the marker approach altogether. Certainly, I think my 4 colleagues this morning have shown you that the development of source tracking methodology is still very much under development. That doesn't mean you can wait. Lord knows how long it's going to take us, but that doesn't mean you can wait. Once source tracking has been applied and you have results, many studies end there. In academia, that's typically what we do. We get involved, we use source tracking, we get results, and then we publish it and we're done. It leaves it up to you to figure out where it's coming from and then how to deal with it. Too few involve going back out and locating the sources, and once you've done that, plus being able to implement BMPs on those sources and then monitor to demonstrate BMP effectiveness. This can be labor intensive, and in my own experience, I've got the 1 8-year study, and we currently have 3 studies ongoing in Virginia, which have been in progress now for over 10 years of continuous funding from different sources. And in a few cases, in some of our beaches, we've solved many of the beach problems. After everything was wrecked by Hurricane Elizabeth in 2003, we found most of the problems, but we still have a few problem beaches in Virginia where, for lack of anything better to do, we've brought out the heavy equipment and we've moved tons of sand, looking for something, looking for anything, and haven't found anything. I don't know where it's coming from. In some cases, I think there's a marine mammal impact from the eastern bottlenose dolphins that are migrating up and down off the coast. But I don't know where it's coming from, and we've been working on those beaches for years. So it can be labor intensive, and years are involved, and many of you have dealt with these very same issues. And so thank you for inviting me. It's a pleasure to come down here and talk science for a full day. I've certainly enjoyed it, and I hope you have as well. So anybody have anything on case studies and your own experience?